Okay, children, welcome you all to the second day of our biology revision. So we were discussing the lesson number 1 of the grade 10, chemical basis of life. Okay, so the previous day you can watch our video. If you do not have watched still, it is better if you can watch the previous video and come back to this video. Because this is continue from our previous lesson. So we discussed the chemical basis of life. We started this lesson. So within this lesson, so far we discussed the various things, right? So in here, basically we focus to this lesson is focused to discuss about the biomolecules, right? You know what are the biomolecules? We were discussed carbohydrates, right? So there are 4 biomolecules: carbohydrates, lipids, proteins, and nucleic acids. So in the previous day we have discussed carbohydrates, right? So you can watch our previous video and come back to today's lesson, today's part. So we have to continue this lesson by a very important part. This part you should have to be very careful. You have to focus on this part because you can score very easily if you can get the knowledge on this identification test, right? So first, what we have discussed, the carbohydrate. So within the carbohydrates, you know there are 3 types of carbohydrates: monosaccharides, disaccharides, and polysaccharides, right? So for an glucose, right? So in here we are going to identify how to identify the glucose. If there is any glucose solution, right? If there is any food type, if the glucose is present, it gives a color. It changes. It gives a color indicator, right? Okay. So first we take a food sample which contains glucose, right? We can get a glucose itself. You know, glucose itself. We do have. We can get itself as the glucose, and we can dissolve it in the water and can make a solution, right? So, but the thing, milky appearance is there. So we have to filter by using a filter paper and get the filtrate. So this filtrate, we can collect it into the test tube. So this filtrate, right, it is a colorless, right? If we directly dissolve the glucose in water, it will be appeared as a milky appearance, right? So, but when we filter it by using a filter paper, we can get the filtrate as a colorless or transparent solution. So first we collect this transparent solution into a test tube. Then what is the second the most important region which we need in order to identify this glucose? It is Benedict solution. Okay, it is Benedict solution. So this is we have to add some Benedict drops of the Benedict solution into this glucose containing test tube. Other than this Benedict solution, we can also use a region called Phelan solution, right? So both of these solutions we can use in order to identify the glucose, because both of these contain the same chemical formula, right? So that copper sulfate, right? So we can either Benedict or Phelan solution we can add into this region. Okay, add into this test tube, right? This Benedict region, we can add 2 drops of Benedict region. We can add to this glucose. Then what is the next important step is, after adding this Benedict solution, we have to heat, right? We have this structure, we have to get heated. So how we can heat? We cannot directly heat it by using a Bunsen burner. We have to immerse it in a water bath and heat. Why we use a water bath? What's the reason for using water bath unless of heating directly? The reason is we can get a simultaneous heat around this simultaneous heat. We can distribute all of this food sample, right? Let's say if we cross it into a Bunsen burner, sometimes some food parts only can be heated by that Bunsen burner, right? Because the heat is not spread evenly to the test tube. But by using this method, what happened? First, the water is heated. So the first water is heated, then from the water, this test tube, the food sample in this test tube will be heated. So it is the reason why we use a water bath instead of direct heating, right? So what is the observation? So it is the most important thing you have to remember. It will give a brick red precipitator like this, right? So precipitate means the solid part, it is floating on the test tube, right? So this is the whole procedure of the test for glucose. So what is the, you have to keep remember, when glucose is heated by by using the Benedict solution and heated that the glucose containing test tube, then it will bring brick red precipitate. Okay, so ultimately how we can identify glucose, if a food sample, if it contains glucose, if we add Benedict solution and get heated, then it will turn into that colorless glucose solution. It will turn into brick red precipitate. So it is the ultimate result of this test, right? So there are so many videos you can watch in the YouTube. Also, I have short video and I will share with you all that in order to what is the ultimate color change of this. You can get an idea of this brick red precipitate by using this video. I stop this. Okay, I think you can see this video, right? This is the color of the brick red precipitate, right? This is the color. That means that solid part, it is floating on this solution, right? You can see this Benedict test. First, we have to get the test tube and hold it by using a test tube holders, right? We do not touch it directly, right? Because we have to heat, right? in here actually, in this video, it doesn't take a water bath, right? In this video, it is heating in the directly. But if we use a water bath, it is very easier, right? This method is also correct, but if we can use a water bath, it will be easier to get this color change, right? Right, you can see. Finally, it is actually convert under various color changes. You don't need to keep remember the color changes, right? So ultimate color change, if the glucose is adding by using this Benedict region, and when heat get heated, the ultimate color change is brick red precipitate color, right? So you have to keep remember that part. Right. So this is the test for glucose. Okay. Next, we will move to the next test. You know, now as the carbohydrate, first we discussed about monosaccharide, how to identify monosaccharide. So glucose is the example for monosaccharide. So what is the region we use for the in order to identify that monosaccharide? It is Benedict solution, or we can use either Fading solution. So ultimate result was brick red precipitate. So now we are going to test the disaccharides, right? So what is the example for disaccharide? You know, sucrose, right? Sucrose is one of the example for a disaccharide, right? So the procedure is same as the test is same as the previous one. The test is same for the identification of glucose, right? So the first part, as the same way, we get the sucrose solution, right? It is a colorless solution. So then what we add? We add Benedict solution, right? And then we heat. So what is the observation that we can get? Actually, there will be nothing happen in that test tube. There will be no remarkable color change happen in this sucrose containing test tube. When we add, when we use this step, it will not make any kind of observation, right? So next, what we are doing? We add. So why actually sucrose doesn't show any reaction for this Benedict solution? You know, sucrose is a disaccharide, and what does it mean? It contains it has made by using two kinds of monosaccharide. What are those two monosaccharides? Glucose and fructose. So therefore, this glucose now, the previous test we used for identification of glucose. But in here, both glucose and fructose is present, right? So what we are doing? Therefore, we have to isolate this glucose one. If only we can, it will show any reaction to this Benedict solution, right? So what we are doing? We add some sulfuric acid, right? So the sulfuric acid, what will happen? Then this glucose, from this glucose, we can get this, from this sucrose, we can only it after adding the sulfuric acid, we can take glucose from the reaction, right? Now from previously sucrose, it present both glucose and fructose, but after adding it, we can get the glucose in the medium. So glucose, you know, it will it is show a color change for the Benedict solution. So what will happen? The previous color change, it also happened in this same test, right? So when we add some Benedict solution and get heated, then what happened? Benedict solution, what happened? It will give a brick red precipitate because it has present glucose in this solution. Did you get that point? Right? You know, sucrose, it is disaccharide. Disaccharide means there are two kind of monosaccharides. They are glucose and fructose. So Benedict solution, it is only give the color change for the glucose, only glucose. So what we can do to, in order to have only the glucose in this region, we can add sulfuric acid or sometimes HCl, or HCl is also correct. But in here, in your textbook, it has given the sulfuric acid. So from the sulfuric acid, we can get the glucose molecules in the solution. Then the glucose is when we are in Benedict solution, then it will give the brick red precipitate as the previous test. Right? I think you can understand this test also. So there are another short video we can watch to get an idea on this color change. Actually, I will show you that. Okay, I think you can see the screen, this video. You can watch this video in this is the sucrose, right? So it will not give any reaction to the Benedict region. Therefore, we have to break down as for the glucose, right? Therefore, we have to add sulfuric acid. Right? When we heat this region, you can see it gives some color changes. Okay? So first, if before adding the HCl or sulfuric, it will not give any color change, right? So then we can add some diluted HCl or sulfuric. Now we add that Benedict solution. You can see it is converting to brick red precipitate, right? Right. So when there is only sucrose, it will not give any color change for the Benedict solution. But when we add some sulfuric or HCl HCl acid to this region, what happens? It will break down to glucose plus fructose, glucose and fructose. So you know, glucose, it gives the color change for the Benedict solution. So ultimately we can get the brick red precipitate. Okay. So you can find out the videos in the YouTube also. Okay. I will share the screen again. So ultimately we can get the brick red precipitate in order to identify this sucrose, right? So first even though we add Benedict solution in the first case, we are unable to get any color change because sucrose is consist with the glucose and fructose. So when there is fructose, it will not give color change for this Benedict solution. So what we are doing, we add sulfuric or HCl into this region in order to this sucrose solution. Then what happens? We can it will break down, right? Glucose and because of this acid, the sucrose is break down to glucose and fructose. Then when there is glucose, it will show color change to this Benedict solution. What is the ultimate color? It is brick red precipitate, right? So you can easily keep remember this color. Okay. Next, then what is then the third type of carbohydrate? It is polysaccharide. Right now we discussed monosaccharides. It's a glucose. Then disaccharide, the sucrose test, and now polysaccharide. So polysaccharide, you know, what is the example for polysaccharide? It is starch. So how we can identify starch? So it is the starch test. It is very easy. We have to, we do have only need iodine solution. So what is the color of the iodine solution? It is brown, right? Brown color. So get the full mixture. Right, let's say we can get any kind of starch content food. We have to dissolve it in the water, but you know the starch is not dissolved in the normal water, so we have to get the hot water, you know, when kind of making this starch containing solution, starch containing food, we cannot dissolve it in the normal water. So we have to get the hot water in order to dissolve that food sample. So we dissolve this food sample in a hot water, and by using a filter paper, we have to filter this food, right? So ultimately, it will give a colorless filtrate, right? So this is the filtrate, it is colorless. So then what we are adding to identify the starch, we can get the iodine region. So it is, what is the color of the iodine solution? It is brown, okay? In here actually it represents blue, but it is actually brown color. So when we add the brown, when we add this iodine to this food sample containing this solution, what is the ultimate color change? It is dark blue or blackish blue. We can explain it in various terms, right? But it is mostly dark blue, right? Blackish colored blue or purple. That means the test is positive. That means starch is present now, right? It is very easy. When the starch is present, what happens when we add in iodine solution? It will turn into dark blue or blackish blue, right? So this is also you have various YouTube videos. You can simply watch this video and get the idea of what is the color of this reaction. Okay, so when the starch is present and iodine, when we add iodine, what happens? The ultimate color change will be dark blue. Okay, right. So with that, we can move to the next biomolecule. What is the next biomolecule? It is protein. Okay, so far we discussed carbohydrates, right? So under that, there are 3 types of carbohydrates: monosaccharides, disaccharides, and polysaccharides. Under that, we discussed what are the elemental composition and where these carbohydrates are, from which places we can found out this carbohydrate contain carbohydrates in our environment. Then we discussed what is the significance of carbohydrates, right? Then we discussed the identification test of carbohydrates. Now we came to the next biomolecule. It is protein. So first we have to identify what are the elemental composition of these proteins. They are carbon, hydrogen, oxygen, and nitrogen, but sometimes it can present sulfur also, right? What are the elemental composition? Carbon, hydrogen, oxygen, and nitrogen, sometimes sulfur. Okay, right. So what is the structure of a protein, right? Actually, the protein is not a single unit, right? It is a polymer. We term as it as a polymer. That means it contains this polymer or this protein has made out of combining with certain units, right? Certain monomers, certain number of monomers. This protein or the polymer it is created, right? So මූලිකම වෙන්නේ කියන්නේ මේ ප්‍රෝටීන් එක හැදිලා තියෙන්නේ අපි කියනවා එක යුනිට් එක එක මොනම එකක් බහු අවයවීකරණයෙන් තමයි මේ ස්ට්‍රක්චර් එක හැදිලා තියෙන්නේ ප්‍රෝටීන්. ඒකට මොකක්ද ඒකේ පුංචිම යුනිට් එක මොකක්ද මොන යුනිට් එකෙන්ද ඒක හැදිලා තියෙන්නේ? amino acid. So the monomer is amino acids, right? So there are nearly 20 amino acids present. So according to this type of amino acids, this protein type will be decided, right? So this is the structure of protein. So when we take a monomer or this single unit, what is the single unit? It is the amino acid, right? So this amino acid, when we look at about the structure of this amino acid, it has created by combining 4 groups, right? First one, it is there is around the carbon there are 4 groups, right? There are 2 groups actually, amino group and carboxyl group, and the next it has R group, right? This R may be any type, and there is another hydrogen atom. So this is the structure of amino acid. So what are the 4 units, right? This amino acid is created by combining 4 units, right? So what are the 4 units? There should be amino group. and there should be carboxyl group, and it has R group. R unit. This R may be changed according to the type of amino acids, and another hydrogen atom is present. Okay, so this is the structure of amino acids. According to this type of R, the amino acid will be decided. The type of amino acid will be decided. So you have to keep remember that. And actually the when this R is present as H, that means hydrogen atom is present as for this R group, we call that amino acid as the glycine. That is the simplest amino acids present in the within the organisms, right? Amino acids, right? Glycine. Glycine is the simplest amino acid present in the organism, right? Okay. The next. Next, we have to identify about the test, right? Okay. Okay. We will move to the so in here, I'm sorry, I haven't included the significance of the proteins, right? Before this one, there should be a part. You have to get knowledge on the significance of proteins, right? So what are the significance of proteins? The proteins can be used as an energy source, right? Like carbohydrates, these proteins can be used as an energy source, right? If it doesn't present, if it doesn't present the a carbohydrate amount in order to get the energy, so we can, so in the body, these proteins also can be used for the energy, to supply the energy. Then the another significance is what? To make structural components in the organisms. What do you mean by structural components? You know, this protein is important for the cell membrane, right? So the cell membrane, it is consistent with this protein. So you have to remember structural as a structural component in the second significant. And so the cell membrane, within the cell membrane, these proteins are present. And you know, in the organisms, in the animals, right? There's a protein called keratin, right? So the animals' feathers, hairs, these keratin proteins are contained. Okay, and the next one is these proteins are in the enzymes. You know the enzymes, right? So what the enzymes are component which is catalyze the biochemical reactions in the organisms. What are the biochemical reactions? That means, you know, in order to digest various food components, right? We do present within the, you know, in the body, in our body, we have a certain temperature within our body. That means nearly 37 Celsius degrees. So within this temperature, there not be actually doesn't happen some reactions in our body, right? Within our normal temperature, some reactions are not happen within our body. So what is the this enzyme do? So when these enzymes present, these chemicals, right, these reactions, they can catalyze. Even within the body temperature, when they present these enzymes, right, so they can catalyze these biochemical reactions. For an example, amylase. So what is it? What is the function of amylase? It converts starch into maltose. Right. In here also, actually, we have to identify this in this test for proteins. We can have to identify this test. So starch, when present this amylase enzyme, it converts to maltose. Okay. So therefore, protein is important as an enzyme, right? And also as hormones. Right, as hormones, the proteins contain various hormones within animal bodies, right? In order to they involve homeostasis and coordinations of organisms, and also they are present in the antibodies, right? In our body, in order to protect from the various microorganisms, the body creates these antibodies. So these antibodies contain which biomolecule? Protein. So these are the significance of the proteins, and you have to remember these 5 benefits or the 5 importance of the proteins. What are those? As an energy source, and it makes the structural components, right? As an enzymes, as hormones, and as antibodies, right? So the next, we have to identify the test for proteins. So first, enzymes. We now know the enzymes are protein. So we can identify this enzyme, and if actually in your textbook this test is there, right? Activity of amylase on starch, right? I explained that what happened when amylase is present, it converts starch into maltose, right? So if we take a starch sample and we add some amylase into this this solution, and then we can take some drops, right, from this region, from this solution, and we take a porcelain tile, right, and we take some drops into this porcelain tile. Then we add some, what is this region? It is iodine, right? What happened when the iodine present? In the previously we discussed, when we add iodine into the starch, what is the color reaction? It will give a dark blue, right? Blue color. So what happened when this starch contain starch and amylase present? Then first actually we take 2 to 3 drops of this food sample, and we add for this porcelain tile, and we add some iodine. Then it will give this blue color, right? And we continue this process within 20 minutes. So what happened within this 20 minutes? This amylase, right? These enzymes, it can break down starch into small parts. That means it break down this starch into maltose, right? So the finally, what is the what is the in test tube? What present? It is maltose, not the starch. At the first it present the starch. Therefore, when we add iodine, then it will give a blue color, right? But after that, right, within we have to continue this by taking 2 to 3 drops of this mixture, and to this porcelain tile within a 2 minutes time interval and during a 20 minutes period. So what happened? Gradually, this starches break down into maltose. So ultimately, it present what? Maltose, right? So that means there are no more starch. So then will it show any color change to this iodine? No, right? Only the color of the this iodine we can observe, right? What is the color of iodine? It is brown. So at the last drop, when we add when we take some drops from this food sample mixture and place into this porcelain tile and add this iodine solution, what is the color? It will not make any color change. It is only appearing the color of the iodine solution. So it is brownish color. Iodine is brown color. So it doesn't show any color change, right? So this when the maltose is present in this food sample, that means it will not show any color change because there are no more starch present in this food sample mixture. Okay, so this is first test you can identify about the test for proteins, and in here we identify whether this enzyme is present or not. Right. The next test is we call this test as biuret test. So what are the reagents we need to perform this test? We need sodium hydroxide and copper sulfate. So this is the biuret reagent, right? So sodium hydroxide and copper sulfate. When we add these two reagents to proteins, then it will give a purple color change. It will give a purple color, right? So when we take a food sample containing proteins and then we add some sodium hydroxide and copper sulfate to that, then it will give purple color change. Purple color. Okay, so let's watch a video how to identify this biuret test. Then you can get a clear knowledge on what kind of color change happens in this process, right? Right, I think you can now see the video, right? Carefully watch it. Now we need protein content food sample. So in here it is used egg. Egg is protein. So we use a egg solution here, and we add sodium hydroxide and copper sulfate. It is the biuret reagent. Then let's see what kind of a color change happen in this process. Okay. Right, as it says that sodium hydroxide, some drops of sodium hydroxide and some drops of copper sulfate we add to the protein contain this test tube, right? Right, so at the top, what you can see, it is color is changing to the purple color. That means we can, from that experiment, what we can identify, this solution or this test tube, it contains proteins. Okay, so with the biuret region, this food sample, it contains proteins. Okay. Now here it has taken some non-protein foods. That means breads, kind of seeds, oils, right? So will they give a positive result for the biuret test? So it is what in this video they are testing, right? Whether will these other non-protein food items give a positive result to the biuret test? So we will look at what happened. Right now, in the first test tube, they do not contain, we can identify they are not contain proteins, right? Right. You can see this in YouTube videos, right? I will share the screen again. Okay. The next we have to identify let's move to the next biomolecule. It is lipids, right? Lipid. So what do you know about the lipids? So we have to first identify what are the elements belong to this group. They are carbon, hydrogen, oxygen. They are the elements belongs to this group, this lipids, right? And what are there are two groups which belongs to these lipids. They are fat and oil. Fat and oil. So fat means what? Fats are the ones which are solid at the room temperature, right? You know, the butter, margarine, right? These are actually these are the solid in the room temperature. And liquids are what? Generally the various oils like coconut oil, ginger oil, right? There are various oils. They are belongs to this. They are liquids in the room temperature, so they are belonging to the oil group, right? So fat means what? They are solid at the temperature. Lipids are two types, fat and liquid. So fat means they are solid at the room temperature. Oil means what? They are liquid at room temperature. So for an example, what are the foods? Coconut oil, ginger oil, ground nuts, butter, margarine. These are some examples. So okay. So now we have to identify how this lipid structure is made of. Right? So there is a glycerol molecule and fatty acids. Actually, there are 3 fatty acids responsible to make a lipid molecule. And within this process, a water molecule will be released, right? So what is how a lipid molecule is made of? By forming of a glycerol molecule plus 3 fatty acids, right? The 3 is not much important. You have to remember fatty acids and glycerols are responsible to make a lipid molecule, right? Okay. Right. Next we move. This is what the structural representation of this lipid, right? You can see this is the glycerol molecule and there are 3 fatty acids, right? These are long chains, right? You can see there the carbon and the hydrogen is present, right? In here actually lipids, you see the elemental composition is same as for the carbohydrate, CH in the carbohydrate also, the elemental constitution was CHO. But the thing is that, remember, in the carbohydrate, the hydrogen and oxygen present as 2:1 ratio, right? But in here, oxygen is less than 1. Therefore, actually the lipids, the 2:1 ratio is not present in the lipids. But the elemental constitution is same as for the carbohydrate. They are carbon, hydrogen, and oxygen. Okay, right. This is the structure of the lipid molecule. So, what are the significance of lipids? Okay, so this is also important as energy source. So, discussed what are the biomolecules responsible for energy sources? Energy source, carbohydrates. The previously we discussed, and then we discussed proteins. They are important as these carbohydrates, and lipids are also responsible for the energy source. And at the same time, these lipids are responsible for the energy source, like carbohydrates and proteins, and to form different structural components. So in the previously, when we discussed about the proteins also, we discussed they are important for a structural component. And also within the lipids also, they are important to form different structural components, right? So like what? Cell membrane, right? All the within the cell, the cell membrane, it contains lipids like phospholipids, cholesterols. There are this cell membrane is the cell membrane structure it has created with this lipids like phospholipids and cholesterol, right? And the next significance is what? For conservation of water, right? In the plant body, there is a wax known as cutin layer. So that means that because of this cutin layer, plant body, they can conserve the water within their body, right? So this cutin layer is important for plant bodies in order to conservation of water. And also, the most animals also, they contain this they also contain wax. So it helps to avoid desiccation, as it is when it is important to conserving of this water, right? And the next one is to maintain the body temperature, right? The birds, mammals, they do not change their body temperature according to the environment. We call them as warm-blooded animals, right? So therefore, within their skin, right, in the they possess in the hypodermis, they possess a fat layer. So fat layer is what it is we call it as a thermal insulator. That means it doesn't change because of this fat layer, they can maintain their body temperature in a certain range. Even though environmental temperature is changed, but because of this fat layer, right, they can maintain a specific range of body temperature. And also, the next one, to protect internal body organs, right? This fat layer, it surrounds many internal organs like heart, right? The structure, this because of this structure, various shocks they can absorb, shock absorber, like it acts as a shock absorber, this lipid layer, right? Therefore, because of these external pressures, they can protect the very important internal body organs. That is important. Another important within this lipid, right? The next one is to synthesize some hormones, right? various hormones like estrogen, testosterone, cortisol, right? These hormones are to synthesize these hormones. These lipids are important, right? So you have to remember these points and to describe one point in a very 2 lines to one or two sentences. Just remember the significance of these lipids as point, and then you can describe it in a 2 to 3 sentences, right? Okay. The next we have to identify the test. What is the test to identify lipids? It is Sudan 3. So you all have to actually remind all this test. Get a short note of all these tests in one place, right? Carbohydrate test, protein test, lipid test, right? You have to take a note in all these tests in one place. Then it is easier to keep remember. So what is the test we can identify the lipids there? It is Sudan 3 test, right? So what is the color change happens when lipids present by using the Sudan 3 region? It will give a pink or red globules, right? Globules, right? It will give when the lipid contains, for example, let's say we get a coconut oil, or we take a groundnut content solution, then if we add Sudan 3, what is the color change? It will change it will appear as pink to red, pink or red globules. Okay, so I will show that this observation. There is a short video. Okay, I think you can see the screen now, video. Sudan 3. First we take 2 milliliters of coconut oil. You can get any kind of oil, it doesn't matter. Okay, actually there is some error in this video. It will not working. You can watch this video in the YouTube also, right? So ultimately, when we take coconut oil or whatever oil and add some Sudan 3 region, then it will give a pink or to red, pink or red globules, right? So I'm sorry, it doesn't play anything. There is some error. I think. Okay, you can see it is in the YouTube also. Right, so pink or red globules you can get by using this within the lipid test. By when we add some Sudan 3, it will give pink or red globules, right? With that, let's move to the next biomolecule. It is nucleic acid, right? So what is the elemental composition of this nucleic acid? It presents carbon, hydrogen, oxygen, nitrogen, and phosphorus, right? carbon, hydrogen, oxygen, nitrogen, and phosphorus. So it is important to identify this elemental composition in every biomolecule. So how then you have to remember there are two types of nucleic acids. What are those nucleic acids? Two types: ribonucleic acids and deoxyribonucleic acids. Right? Ribonucleic acids and deoxyribonucleic acids. So this nucleic acid, they are actually a polymer. That means one unit is combining with various other units and make a polymer, and then we call it as a nucleic acid. So what is the monomer of this? If any two of this nucleic acid, is it? It is nucleotide, right? So this nucleic acid කියන්නේ එක polymer එකක්, බහු අවයව එකක්. ඒක හැදෙන්නේ එක unit එකක්, monomer එකක් බහු අවයවීකරණය වෙලා. එතකොට single unit එක අපි කියනවා nucleotide එකක් කියලා. So there are two types of nucleotides according to the sugar group. Right now, you know this is a nucleotide, and in this nucleotides there are 3 units. What are the 3 units? There is a phosphate group and 5 carbon sugar and nitrogenous base. Right? So according to the type of sugar, right, there are 2 types of sugar, ribose and deoxyribose. So according to this type of sugar, the nucleotide will be decided, right? What is how actually there are 2 types of nucleotide present? Because the difference of this, the variation of this 5 carbon sugar, right? When it present ribose sugar, we call this nucleotide as ribonucleotide. When it present deoxyribose sugar, we call this nucleotide as deoxyribonucleotide. So when by polymerization of this ribonucleotide, then it will make ribonucleic acid, and the polymerization of deoxyribonucleotide, it will it creates deoxyribonucleic acid or DNA, right? This ribonucleotide, it will make ribonucleic acid, that means RNA, and this deoxyribonucleotide, by polymerizing with deoxyribonucleotide, it will make DNA, right? Deoxyribonucleic acid, it is mean DNA, right? So this is the structure of RNA. So what is the why there are 2 types of nucleotide present? Because these variations of this sugar group. So when it contains ribose sugar, we call this nucleic acid as the ribonucleic acid, right? So this is a single strand, right? There is a single strand. This is a single strand. This RNA is a single strand. Only one chain is there, right? But you can see DNA, right? So the DNA is how this RNA DNA is we can demarcate by using because of this sugar group. So in this DNA, it contains deoxyribose sugar, and the deoxyribonucleic acid, it has 2 strands. And you can see we call this structure as a double helix structure, and this structure has discovered by 2 scientists named Watson and Crick, right? So you have to remember this RNA is a single strand and the DNA is 2 strands. Okay, right. So what are the significance of this nucleic acids? So these nucleic acids, they are responsible for storage of genetic information, right? All the genetic information responsible for organisms, like let's say for your hair, for the hair growth, for the tall and the short, right? Your hair color, skin color, right? Any kind of your appearance, right? It will decided this by according to the genetic information which is storage, according to the genetic information which is stored in this nucleic acid, DNA and RNA. Right? All the everyone they do have various unique characteristics. So that how this characteristic is decided according to our genetic information. So from where this genetic information is stored in the nucleic acid called RNA or DNA, right? And the next is these nucleic acids, they can transfer genetic information from generation to generation, right? They are not only stored in the nucleic acids, right? They are not only stored in, they can transfer, this genetic information can transfer from generation to generation. Is also a significance of nucleic acid. And the next is they are important in protein synthesis, right? So the nucleic acids are also important in the protein synthesis, and they are control all cellular activities in a cell, right? So these are some new benefits or the importance of nucleic acids you should have to keep remember. Okay, they are stored. What are the significances? They store genetic information within DNA and RNA, and this information can transfer from generation to generation, and they are important in protein synthesis, and they can control all cell activities, all activities in the cell. Right. So this is a picture of this DNA and RNA. Right? Okay. So today we discussed 3 types of biomolecules, and from today we have finished the remaining biomolecules. Right? So we discussed lipids, right? Lipids and first we discussed the proteins. Right? The proteins are contained with carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. And it is also a polymer. That means it made by combining with various monomers, right? So the monomer is amino acid. So amino acid contains 4 units. It is amino group and carboxyl group, and R group and hydrogen atom. Right? The simplest amino acid we call it as but glycine, because this RS is replaced by a hydrogen atom. Okay. Then we discussed some what are the significances of the proteins, like it as an energy source, as a structural component, and it contains in the enzymes, hormones, within the hormones, and as antibodies. So then we discuss what are the tests having for identify for proteins. So we can identify the test. If we can identify the test in our enzymes, right? If we can get a positive result for this enzymes, right? This enzymes test, then we can say these enzymes contain proteins, right? What is the test we use? We get a starch and amylase. Amylase is an enzyme. So what does it do? It breaks the starch into small component called maltose, right? So what happened when what is the region we are using to identify this amylase on starch? It is iodine, right? Iodine solution. So we can add iodine at the first. We can it will give a color change, because at the first it in the solution it has starch, but in the latter part of this process, it is gradually break down into maltose. Then finally we cannot take the since there are no more starch present in the solution, we can't get the blue color or the color change to this iodine test. Then there is no, it will not show any color change. It will give the brownish color, which is the color of the iodine, right? Then another test is there called biuret test. So any kind of a protein, it will make into a purple color of this when we add this two regions, sodium hydroxide and copper sulfate. Then we discuss about the third biomolecule, which is lipids, right? The lipid is also contain CHO, like the carbohydrate, but the thing is the oxygen is very small. The present of this oxygen is less than the carbohydrate, right? There are two groups we discuss: fats and oil. Fats means solid at the room temperature, oils mean the liquids at the room temperature. And how a lipid molecule is created by combining 3 fatty acids and glycerol, right? So we discuss these things. So then there is a test to identify lipids. They are Sudan free. So it will give a pink or red globules, right? So we discuss the significance of the lipids. So we can use them as an energy source, different structural component like cell membrane, and for conserving of water, right? In the plant body, they do have cutin layer of a wax called cutin layer. In the animal body also, they have, and they maintain the body temperature, right? So because of presenting this lipid layer on our skin, we can regulate. We means the warm-blooded animals like birds, mammals, they can regulate the body temperature and protect internal body organs, because this fat layer, it acts as a shock absorbent, right? So they can protect the internal body organs and to synthesize some hormones. So these are the significance of lipids. And finally, we discussed the final biomolecule, which is nucleic acid. And we discussed the elemental composition here, right? And in here, you have to remember there are no any tests to identify nucleic acid, because we cannot perform it in the living state. You know the nucleic acid. We have to if we have to test this nucleic acid, we have to use a living organism. So we do not have any test to identify at this level, right? So we discuss what are the elemental composition. They are CHON and P. So there are two types of nucleic acids, DNA and RNA. How this DNA and RNA is created? These are also, polymers, right? So they are created. They are made of by combining monomers, right? So the monomer is nucleotide, and RNA is made of ribonucleotide, deoxyribo. The DNA is made of deoxyribonucleotide, right? Okay. So these things we discussed, and finally we discussed what are the significance of nucleic acids. So all the genetic information, they are stored in this DNA or RNA, and this genetic information, they are transferred from generation to another generation, and they are important in protein synthesis as well as control all cellular activities in cell. So these are the today with that we finished all the 4 types of biomolecules which we had to discuss within this chapter, right? First lesson, carbohydrate, protein, lipids, and nucleic acids. The next there are two topics we had to discuss: water and vitamins. So in the next day we will discuss those two topics, and we can finish the lesson by the next day, and then we will discuss some questions that has asked in the past papers in the exams, right? So I think you may have get an idea. A quick revision. Actually, this is not a theory class. We are doing a revision class. That is why I speedily quickly revised you all in this lesson. So with that, we can finish the lesson for today. And thank you very much for us today.